The overall goal of the following experiment is to evaluate the presence of protein-protein interactions on the cell surface. This is achieved by cloning two receptors of interest amino terminal to two fluorophores that have overlapping emission and excitation spectra. As a second step, the two vectors are simultaneously transfected into endothelial cells for receptor expression. At this point, the fluorophores aid in evaluating the overall extent of expression as well as receptor localization. Next, the imaging parameters are established using single transfectants as a control on the confocal microscope. Individually transfected cells are selected based on their relative levels of receptor expression and localization. Regions within these cells are then imaged in order to evaluate the extent of FRET. Results are obtained that show Tie 1 and Tie 2 interactions on the endothelial cell surface based on FRET and confocal microscopy.
Hi, I'm Tom Seegar from the laboratory of Dr. William Barton in the Department of Biochemistry and Molecular Biology at Virginia Commonwealth University. Today we will show you a procedure on evaluating protein-protein interactions in endothelial cells by a FRET-based confocal microscopy. In our lab, we use this to study receptor interactions during angiogenesis. So let's get started.
To image protein-protein interactions using a FRET-based proximity assay, begin by cloning your receptors of interest into the NHE1 EcoR1 sites of the pcDNA 3.1 hygro or neo vector containing monomeric CFP or YFP. The determination of FRET is empirical.